Thank you, Nanopore, for having me here today. I'm going to talk to you about a little bit the work we've been conducting on pediatric germ cell tumors. So, you might know, pediatric cancer is a rare event, and germ cell tumors are even more rare, comprising around 3% of all the pediatric diagnoses. Um, and they are a group of heterogeneous tumors that arise from primordial germ cells that undergo genetic alterations. And based on the different differentiation stages of that cell, it's going to originate different subtypes of this tumor type. So they normally occur in the gonads, like ovary and testes, but they can also occur along the midline of the body, and their diagnosis is normally performed with a pathology report from a biopsy and can be corroborated with some serum markers like alpha fetoprotein, beta HCG, and LDH. So here we aimed to explore whether nanopore sequencing, RNA sequencing, could give us gene expression patterns specific to each unique subtype of germ cell tumors, and whether we could use that to a machine learning algorithm to classify those subtypes. So we used samples from 47 pediatric germ cell tumors, and we had 20 samples from cryopreserved tissue and 36 samples from FFPE, because we had like tumors that were stored both as cryopreserved and FFPE. So we sequenced them both. So we had a total of 56 samples, and this is like the distribution based on their subtypes. To do that, we used the PCB109 kit in the MinION flow cells. We also multiplexed them at around 4 to 10 samples per run, and we did that for around 12 hours. So we got around 200K reads per sample, and we had an N50 of 328 base pairs for cryopreserved samples and 205 base pairs for FFPE. We performed base calling using Minao and Dorado in super high accuracy mode, did alignment and gene expression counts using Minimap and the ensemble transcriptome. We performed a PCA analysis to inspect the data, and then we started doing the machine learn pipeline that was developed by our group here at UNC Chapel Hill, that was previously published for pediatric acute leukemias and more recently for some solid tumors as well. We used the partial least squares regression and support vector machine method, and we also used the leave one out cross-validation approach. So to inspect the data, we ran the PCA with all of the transcriptome data. And interestingly, instead of like getting a separation based on their subtypes, we got a separation based on their storage. So we got here in green all the samples that were from FFPE, and in pink all the samples that were from cryopreserved tissue. So based on the literature, we decided to make this, try to get a subset of genes that could more distinguish those subtypes. And so these genes were gathered based on being previously reported as altered or have a higher or lower expression in some subtypes. So we use that for the next analysis. So here we got more clear separation of those subtypes. In blue, we have the dysgerminoma samples. In red, we have our yolk sac tumors. And in black, we have all the teratomas, because like they didn't differentiate it much here if they were like immature teratomas or mature teratomas. We also did this heat map to show the expression profiles. The yolk sacs had a higher expression of RBB4. Dysgerminomas showed a higher expression of PAL5F1 and NOG as expected. And the teratomas cluster had a higher expression of TP53 and KRAS. So then we started using our data to train and test our previous pipeline of like the machine learning. So we started with 48 samples because we excluded all the mixed cases. Um, and to handle the low coverage and the sparsity of the data, we excluded the genes with 0 counts across all samples. So we tried some cutoffs, and the one that got us the best results had like 50% of the non-zero counts excluded, which got us around 11,000 genes. And this was our first attempt of like classifying them. We got 72.9% accuracy, but here I'm not referring as accuracy any threshold. I'm just considering if the sample got correctly classified or incorrect. Um, so we see here also that the prediction probabilities are consistently below zero point 0.8, so there's a lot of room for refinement. So in order to improve that, we did a few things. First thing we did was we got 4 of our mixed samples that had more than 70% of one specific subtype, and we consider them as that major subtype to increase our number. We also grouped the teratomas into one single class. And instead of applying the non-zero counts, the filter of the non-zero across all samples, we decided to implement it within each subgroup. And we got the threshold of 10%. So that got us 6,000 genes. And in this classification try, we got 9.4% accuracy. So we got like way better than the first try, but also we still have some room for refinement. We can see here that 4 out of the 5 samples that were incorrectly classified are from the embryonal carcinoma subtype, which we only had 4 samples. So we can conclude that with that few samples, we are not able to train the model to like be able to classify it. And if we consider the 3 subtypes that we had more than 10 samples to train our model, we got a 97.9% accuracy. So we can conclude that nanopore sequencing represents a potentially globally accessible technology for the characterization of pediatric germ cell tumors, and that our currently classifier was able to classify those major subtypes even with shallow RNA sequencing depth. As for next steps, we are working on some probability calibrations to improve our confidence and like increase those prediction probabilities. We also want to develop a new classifier only for the teratomas because clinically speaking, we also want to differentiate between mature and immature teratomas, which like right now we're not being able to do that. And also increase the number of samples, which is like a challenge since due to the rarity of this tumor. So I want to thank everyone in our research group, especially back home at Barretos Cancer Hospital, also our group here at UNC Chapel Hill, and our partners at St. Jude Children's Research Hospital, and our financial support.